We'll be begin a look at the cell today. And so we'll start by looking at the cell structures themselves and see what we can see inside the cell and outside the cell and go from there. So cells themselves begin with the cell theory. And so cells themselves, as far as humans are concerned, as well as animals of other kinds and plants, they are the building blocks of all of them. And so this is where, in a sense, the whole structure itself relies upon not just the individual cell per se, but all of the cells being able to work together to do all of the different functions that that particular organism needs. And so we're going to have different kinds of cells that are doing that function throughout the way. But in the very beginning, each cell began from a single cell. And so every new cell comes from the division of another cell. And so whether we're replacing cells, like we have here, where we're going to go from a cell that divides up and creates new cells themselves and then sends them along the way, or whether it's being from the original cell in a sense, which we'll talk about in just a second, for that individual cell to create the entire body itself. It's at the cellular level where essentially all of the physiology of the body occurs. And so within the cells, between the cells themselves, this is where all of that physiology is occurring. And so the cells are working to bring things into them, working on those structures inside there, doing things to them, and then getting rid of things. And so we can almost think about the individual cells as being kind of like a whole city. And so for the whole body, we've got individual cells that work at different locations, just like we would have in like a small city. We have different jobs, we have different organizations, and each one of them has their own specific function. And all of those functions, in a sense, are working towards the overall keeping of the body in homeostasis. And so kind of that utopian for the entire body itself, all the cells themselves are working towards that. And as I said, we begin from just a single cell. And so our single cell becomes our zygote. And so it begins from our fertilized ovum or egg cell. And that becomes fertilized from the sperm. And the sperm enters into it. You can see the head piece of the sperm entering into the cell there. And as it starts to come into the cell there, it's going to leave just its genetic material into the inside of the cell. The tail is going to fall off, and then that cell divides. And then that cell, now that we started out with that zygote that was there, it divides and turns into 2 cells, and then 4 cells, and then 8 cells, and so on. And they keep dividing and dividing and dividing, and we get more and more cells. Early on along the way, those cells then have to go through differentiation. So they're going to have to start to divide into different types of tissues. And so we're going to have our 4 basic types of tissues. We have our epithelium, connective tissue, muscle, and then nervous tissue. And depending upon, in a sense, where they are within that mass of cells and the type of cytoplasm that they're accustomed to, in a sense, that's going to dictate which part of the genetic code gets turned off and which parts get turned on. And that's going to give us this differentiation into our 4 different major tissue types. And then from there, those are then going to differentiate some more. So those are then going to differentiate into the different components of epithelium, connective muscle, and nervous tissue as well. When we look at an individual cell, essentially there's 4 major things that every cell is going to have within it. And so one of the more important aspects of a cell is that it has this plasma membrane. And so in our cells, we have a plasma membrane there. That plasma membrane is going to basically keep everything that's inside the cell inside there and keep everything out. And so it's not going to allow for things to come in unless, in a sense, it wants them to. And so it's going to be very selective to allowing certain substances to make its way through the cell. Inside the cell, all of the kind of empty space here is what we call the cytoplasm. And so the cytoplasm is that fluid space that goes all the way from the plasma membrane to the nucleus. And so it's everything contained within that space that's there. And it's made up of fluid, cytosol, And that fluid has floating inside of it different types of organelles. And so we have different organelles that are inside there that are doing different jobs, different functions within the cell. And then on the very inside of the cell, we have a nucleus. And so somewhere buried inside the cell, we're going to have the nucleus. This is going to have all of the genetic material, not just for the cell, but basically it has the entire genetic code for the entire body. And so whether this is a skin cell, a muscle cell, a liver cell, all of them contain all of the DNA for the entire body itself. It's just that it can't look at all of the different code that is there. And then lastly, the thing that we have are ribosomes. So ribosomes are going to be found in different areas within the cells, depending upon the type of cell that we have. But ribosomes' job is to make proteins. No matter what it is, a cell has to be able to make proteins. And whether it's to fix the cell wall, the plasma membrane there, whether it's to add for substances that it's going to need for inside the cell, whether it's making things that are going to come out of the cell and get packaged up, it needs to be able to make those proteins. And that's where ribosomes come into play. So here we have kind of a generic eukaryotic cell or animal cell that we have here. And this has essentially all the different kinds of organelles in the cytoplasm that we could have. So we don't necessarily have a cell that looks just like this per se, because this one is kind of the idealized type of cell. So this has all of the different structures that are there. But it has those four basics as well. And so it has the plasma membrane. It has the nucleus. It has all on the inside here, we have all of the cytoplasm that is inside there. And then lastly, we can see all of the little kind of darker dots that are inside there. We have ribosomes. In this particular case, those ribosomes are actually on another structure, and that structure that they're on is part of the organelles. That is our rough endoplasmic reticulum. So all of this space in here with all of the dots on them is all part of our rough endoplasmic reticulum. In some cells, they also have a smooth endoplasmic reticulum. When we have those structures as well, the endoplasmic reticulum, we're typically going to have a Golgi complex for them. We can package things up and we can have things like lysosomes and peroxisomes within the cell. We have structural components, things like microtubules, microfilaments inside the cell. And so we can help to hold the shape of the cell and maintain the overall shape of that cell. Depending upon the type of cell, those cytoskeleton components are going to vary depending upon the cells themselves. And then one of the more important structures that we have here is mitochondria. And so we have multiple mitochondria in here. There's another one, there's another one, there's another one. A couple more down here that we can see. And those mitochondria are the energy, the source of energy for our cells themselves. And so this is where we're going to get the energy coming from for the cells and have the ability to power the cells and let them do all the different things that they need to do. Certain cells, like the sperm cell, are going to have things like flagellum on it. And so it's going to have a tail or a flagella on there. And then other cells have what kind of looks like just little short stumpy tails. We have cilia. Flagella are going to be able to move the entire cell, and cilia are going to be able to move just substances on their surfaces. So lots of different types of things that we can find within that cytoplasm within the cells themselves. And so cytoplasm is that space between the plasma membrane and the nuclear envelope. So it's in between the nucleus and the outer shell, so to speak, of the cell that's there. It is liquid, so we have cytosol. So we have our fluid portion that is there within that. The majority of it is water, and so we have a large component of the cytoplasm itself is just simply water. But then we also have all of the different organelles that are there. And so we have a huge amount of organelles that are inside there. That water isn't kind of like just having water in a cup or something like that. It is held together by some other special proteins that are inside the cytoplasm, and that makes the cellular makeup inside that cytoplasm to be kind of like that of a gel. And so it's going to be not quite totally fluid, but not quite totally solid either. Somewhere in between, having almost like a jello or something like that, that is almost hardened, but not quite. It's still kind of liquidy and we can move things through it. In the very center of the cell, typically, we have the nucleus itself. And so the nucleus here, we have typically only one per cell. We'll see as we look later on at different types of tissues in the body that we can sometimes have more than one nucleus, but that single nucleus is there when we have a single cell. And so where we have cells that have multiple nuclei, those cells have fused together. That's there. And so usually the nucleus is the largest thing. If we were to look under a microscope, typically the nucleus is what we're going to be able to see inside the cells. All the other organelles we're not really going to be able to see inside the cells itself. And inside there we can see all of these little ribbons that are there. And all of those little ribbons are the chromatin that is inside the cell. It contains all of our DNA, and so it contains that entire blueprint or the entire book. However you want to think about it in terms of the DNA, it's all there. So no matter what kind of cell this nucleus came from, it contains all of the DNA for the entire body itself. But when it comes down to, in a sense, reading all of that blueprint or all of that book, there are portions that it just can't read. And so it can't look at those portions because it's no longer a stem cell kind of a cell. And so it doesn't have that ability to read them. So any cell that we have in our body, for the most part, the most of them are all somatic cells, but we do still retain some stem cells as well. And so it is a relatively large structure inside there. And we see that it is also connected to our rough endoplasmic reticulum. So it is, in a sense, continuous with that rough endoplasmic reticulum, and it's going to allow for it to move substances in and out in terms of our RNA. The nuclear envelope itself is pored, so it does have pores on it. But the nuclear envelope is a little bit different from the plasma membrane of the cell in that the nuclear envelope is very selectively permeable, whereas the plasma membrane is more of a semi-permeable membrane. It's going to allow certain substances in and out of the cell as long as they're permeable through that membrane. Versus for the nucleus, it's going to be very highly selective as to what can get through those pores and into the nucleus itself. And so we can see that the nuclear envelope is a solid structure there with the pores associated with it, the rough endoplasmic reticulum. So rough endoplasmic reticulum, or rough ER, that we have here is a series of little sacs, and so they're kind of interconnected. We can kind of follow our way through kind of like a maze as we're coming through them. And depending upon the portion, this one would probably go down a little bit and then make its way out and so on. And through that rough endoplasmic reticulum, this is where we're primarily going to produce proteins. And so we can see that we have all of these little ribosomes on here, part of the reason why we call it rough endoplasmic reticulum. And so since we have those ribosomes there, ribosomes are in the cell for protein production. So that's what our rough endoplasmic reticulum is going to do. In certain cases, it does produce some lipids as well for that continuous kind of structure, but those are going to occur over in the smooth endoplasmic reticulum. So the smooth endoplasmic reticulum, as we can see, it's devoid of any kind of ribosomes. There's none there. There's nothing sitting on the surface of the cells. There's no studded ribosomes there. So it's not going to be able to produce proteins. But smooth endoplasmic reticulum is going to have many, many different jobs depending upon where it is in the body itself. And so depending upon where we find it, that's going to dictate kind of what that smooth endoplasmic reticulum can do. In the beginning part here of our physiology, the first place that we're going to see smooth endoplasmic reticulum is going to be the muscle cell. And so in muscle cells, the smooth endoplasmic reticulum is going to store calcium. So it's going to work in storage in those cells. Other places it produces stuff. Other places like the liver, it does detoxification for things there, as well as the lipid production at the same time. So these tubules that are there allow for us to have substances move in between them. And as they move in between them, they're running inside the little lumen that is there. And that's going to eventually make its way out to the very outer aspect, at which point it's going to release a vesicle. That vesicle is then going to move off and go to get modified later on in the Golgi apparatus, or the Golgi there. The smooth endoplasmic reticulum, as I said, it's there for dealing with things like detoxification in the liver, storage of calcium in muscle cells, and so that's some of the things that it does. Other places, it's going to, again, back to the liver, we're going to deal with carbohydrates, lipids, steroid hormones for their production, especially when you go down into the reproductive tract for the steroid hormones themselves. And so it really kind of varies as far as its function depending upon the cell that it's in. But it's not going to be able to do all of it, only what's particularly coded for in that particular tissue that we find it. So when we come from the rough endoplasmic reticulum, we have a little vesicle here that leaves the rough endoplasmic reticulum, and then it moves over to the Golgi complex, or the Golgi apparatus. As it makes its way there, the Golgi complex is going to receive our little vesicle, and the side where it's receiving that little vesicle is going to be the cis face, and then the other side is the trans face. And so it basically moves through it, and as it does so, the Golgi apparatus is going to take those substances and it's going to modify them, it's going to alter them, it's going to change what we can find inside of them. And as it's making its way through here, by the time that it gets to the outside, it's been, if it's a protein, it's been sorted, it's been concentrated, it's possibly been modified as it's gone through that Golgi complex that's there. And now we have this vesicle that can leave that trans side. And maybe that vesicle becomes something like a lysosome. Perhaps it becomes a vesicle containing some sort of substance. So maybe it's an endocrine organ and it's created its hormone that's there. Some of those vesicles may eventually leave the cell itself. So they may be destined for exocytosis that are there. And so we've generated this final product that's been packaged up there. In a sense, it's kind of like that of a plant where we've made a substance. We started out making it over here in the rough endoplasmic reticulum. And then as it went through the plant, we needed to potentially modify it a little bit. We need to get all of that particular product all together so that it's all eventually in the same box. So we want to ship all of the same product in that is similar to one another or the same as one another and get them all going to the same place. So we put them all in the same box that's there. And so then we send it from that point on and it can go do its final job, whatever it was that it was meant to do at that point. One of those packages that we package up are lysosomes. And so lysosomes contain digestive enzymes. And so these digestive enzymes that are there for breaking things down. And so in our bodies, we have them as lysosomes in our white blood cells. And so we're going to have a lysosome that's there. Here we have a particular organism that's there, and it is eating a food particle. But even if this was a white blood cell, it would be doing the same thing, except this food particle would potentially be, say, a bacteria. And it brings it into the inside of the cell. It has its lysosome there ready, and those digestive enzymes are going to mix with the phagosome. We get a phagolysosome. The contents come together and mix together, They digest it. If this was a bacteria, it would now be destroyed, and then it sends it out of the cell via exocytosis, and we get rid of that substance at that point. Now it can't harm us anymore. In other instances, those lysosomes may be there for, say, this little green guy here is an organelle. Perhaps it's an old organelle that has been worn out, it's no longer functional, or it's losing its functionality, and the cell is then going to use those lysosomes to break down our organelle, and the same thing, get rid of it as waste out of the cell itself. That is there. And so the ability of those lysosomes is primarily to break things down. So other places, we'll talk about in bone, where in bone, some of those lysosomes are utilized as, especially their low pH, they have acids in them, to break down bone, to dissolve some of the mineral content that is inside bones as well. We also have peroxisomes, and peroxisomes are somewhat similar to that of a lysosome. And so we have this situation where now we have another little vesicle inside the cell, but this vesicle now contains, rather than enzymes, it's containing substances that are going to be able to neutralize problem molecules inside the cell. And so what we have here with peroxisomes is we have our antioxidants. And so the antioxidants are inside there. And antioxidants are then going to go out and potentially get rid of, I'll make it red, something that has either too many or too few electrons. Essentially, they have unpaired electrons. And so they're going to be able to carry out these oxidative reactions. And so they're able to neutralize the potential problems that are inside the cell that are there, where we have molecules that have either too few or too little of those electrons in their outer shell, and our breakdown occurs by either accepting or donating them in that point. So we're able to basically neutralize toxic materials, break those down, get them into something that is no longer harmful to the cell that's there within the cell itself. We're going to be able to neutralize substances that are within the cell, break down fatty acids, amino acids within the cells themselves, and be able to liberate them out into the rest of the body so that we don't get any harm from these substances. Mitochondria are the workhorse, in a sense, of the cell. So this is where we're going to generate all of the ATP for the cell. And so when it comes to energy, we typically think of energy coming from things like the things that we eat. And so we have a sandwich, we may have some carbohydrates in there, we may have some lipids in there, and we may have some proteins in there. From those carbohydrates, lipids, and proteins, we're going to need to be able to extract the energy kept in their bonds. And so in order to do that, we need to make ATP. And that's where the mitochondria comes into play. And so the mitochondria are going to be able to manufacture all the ATP that those cells need. And so they're going to have this ATP there. This is going to be our universal energy source. And this is one of the things that we're going to talk about next in the next series, as we look at the metabolism of the cell itself and kind of the physiology of how the cell actually functions on those. And so the mitochondria is also unique in that it contains its own genetic material, and that genetic material is also maternal, meaning that when we have the egg and the sperm, as they come together, the ovum has the mitochondrial DNA, the sperm cell does not. And so all of that DNA that's on the mitochondria comes from maternal DNA. That's there. And it then has the ability to manufacture its own ATP. There are, but this particular picture doesn't really show it, there are ribosomes there as well, so we can manufacture some proteins as needed also. In order to maintain the shape of the cell, and so when we look at different types of tissues, we see that we have everything from kind of cuboidal cells to columnar cells, taller than they are wide, and then we have squamous cells. And so each one has its nucleus inside there, and it's kind of floating around inside the tissue itself, so to speak. And so those different kinds of cells need to have different types of cytoskeleton in order to maintain their overall shape. And so we can have different types of them. We have microfilaments inside cells. We have intermediate filaments. And then we have microtubules. Each one has a slightly different job, has a slightly different function within the cells themselves. Some cells may have more of one versus the other, all depending upon what that cell is designed for. Here we can see that in some cases, those filaments themselves, the cytoskeleton, is allowing us to transport certain substances along the way. And so we can use them as along with motor proteins to transport things like even vesicles or other proteins or organelles into different portions of the cells themselves. So microfilaments, as their name implies, are very small, and they have a special protein called actin. And so this actin protein that is there, we'll see actin once again when we get into the muscle because it's one of the motor proteins that are involved for muscle contraction. And so they're able to be utilized to where we can, as we move along the actin and along that microfilament, we can move substances from one place to the next. And so we can polymerize them very quickly and allow them to reform very quickly as it goes, which means that we can use them for things like flagella, we can use them for cilia, we can use them for changing the overall shape of the cell itself, as well as holding and maintaining that cell shape rigidly when we need to also. Intermediate filaments are kind of the next up in size, and so these become a little bit larger than what we had before. These ones are more of along the lines of maintaining the structural support of the cell itself. And so these have multiple kind of strands that come together, kind of wound together like a rope or like a large cable inside the cell. As a cell should happen to be kind of pushed down upon in a sense. So if we try and push down on a particular cell, it's going to maintain its shape. So we're not going to be able to squish that cell any as it kind of comes down. The intermediate filaments as well, we have the nucleus of the cell. We're going to see intermediate filaments kind of anchoring the nucleus inside a cell. So they're going to be holding it in place, as well as we saw in that very first picture, we can have other organelles kind of attached onto them and holding them in place as well. The largest of the three are our microtubules. And so these microtubules are essentially hollow tubes. Think of them as being literally a tube that is there. These ones, once again, are going to resist compression. It's kind of like having maybe like a bamboo stick or something that is there. They're also going to be used for movement once again. And so we're going to be able to move substances in and out of the cells along these microtubules. For cells that are going to divide, we're going to be able to move chromosomes along microtubules. So we have to have microtubules in order to get the chromosomes to move from the kind of the central part of the cell out towards the poles of the cell as it starts to divide. Cells need to be held together. And so as we hold cells together, we need some connections between them. We don't want the cells to just kind of float off or fall off and go off in different directions. So we have these special connections between them. And depending upon the type of organism that we're looking at, we're going to see different types and different functions for each one that is there. So the tight junction is essentially one that creates it so that there is no way that anything can get in between the cells. It's almost in a sense kind of like welding the two cells together. And so we don't want anything to move in between those cells and get in between those cells at those points there. So where it comes together here is almost like spot welding the cell together. And so we're going to have them oftentimes all along the surface of those cells when we want to be able to maintain a very tight boundary. And so we're going to see this with things like our skin cells. We're going to see these with things like our digestive tracts cells that are there, all the while being able to basically maintain that structure so that nothing's able to get in or out in between the cells themselves. So very strong connection between them. Desmosomes allow for a space in between the cells. So it allows for that space to go in between the cells. We can move substances in and out of a particular tissue in between those cells that are there, that allow them that little bit of space that's there. It also allows the cells to kind of stretch in between the two. So it allows them to kind of pull apart from one another and then bounce back on those. We see these in our cells, them as well, and other animals. Here we have those connexons that are in between the two, and those connexons are going to, as we pull and stretch on the cells themselves as well, they're either going to be opened or closed as we close and open the space in between the cells themselves. Gap junctions take kind of the next level, so to speak, in that we have those same connections in between the cells, and we're going to allow for substances to move in between the two cells. And so this is going to allow us to move a substance in between them. We'll see these when we get into muscles as well, in that the gap junctions allow us to create a space in between the cells where we can move substances between one cell to the next cell and move them along to the next cell there. So we'll use them in muscles to be able to allow us to move calcium in between one cell and the next, so that we maintain our normal calcium concentrations there on those. So that space that's there, important for muscle contraction. It allows other substances as well to move through, besides calcium as it goes through there. And so water is going to get through. Other small ions are going to be able to move through in between the two cells themselves, and make its way from one cell to the next. And we're going to essentially allow that those gap junctions are going to allow us to be able to move substances from one cell to the next, to the next, to the next as it moves through the tissue itself. And so we're going to be able to move that calcium along. And then when it comes to muscle cells, we're then going to also move an electrical charge in and along the cell as that happens at the same time. And so this allows us to essentially connect the two cells together, allows us to create this junction between the two sides of the cells themselves, and allows us to limit the amount of structural components that might be simply in the cells themselves. Later on, we'll talk about the actual movement of substances through the plasma membrane and how that occurs.